So I am Frank LaFurla. I'm the Dean of the Ayala School of Biological Sciences, and welcome to our distinguished lecture. I'd like to start off by thanking all the people who put in a lot of time and effort. As you can imagine, it takes a lot of hard work to make these events successful. So special thanks to members of the Dean's Office, particularly Shannon, Lizzie, Wes, and also Benedict. Before I begin talking about the formalities, I just want to remind everyone that our BioSci Starbucks is finally open. It only took 4 years, but I hope to see you there. Thus far, it seems like it's a big hit, at least with the students, and so pick your times accordingly, because about 10 before the hour, it seems to be very crowded when classes let out. So today we have, I think, a very special treat, not only because we get to hear from a world-class scientist, but also from an individual who is a world-class photographer. So let me give you a little bit of background about Ian Parker. Many of you know he's a professor in the Department of Neurobiology and Behavior, and he is an expert on cell imaging. He has been working on this area for a long time and has made many seminal contributions to not only basic mechanisms of cell signaling, but also towards understanding how, when these mechanisms go awry, they can lead to disorders like Alzheimer's and autism and multiple sclerosis. He is a very distinguished researcher. He has published over 200 manuscripts, and he is an elected member of the Royal Society of London. He has received a Merit Award from the National Institutes of Health for his science, and he is a very highly sought-after collaborator. He is technically very sophisticated in his spare time, likes to build instruments, like simple things like two-photon microscopes. And also, he is an avid athlete. So many of you might not know this, that he participated in the Badwater Marathon. And he described this to me a couple years ago and was telling me that he was going to run through Death Valley, I think about 150 miles or so. And the way Ian prepares for this is he puts a treadmill in his closet as well as a heater, a space heater, so he could emulate the temperatures that are up there. And so I thought, wow, that was really impressive. And so I asked him, well, what do you get for this? And he shows me his belt buckle, and he goes, well, I get this belt buckle. And I'm like, that's it? So we have a slightly different approach, I guess, toward what's fun. But he is just a really outstanding scientist. You'll see from some of the images, both scientific and his natural photography, that he has a great eye for artwork. And so I think it's a great honor that we have him here. And before I introduce him and welcome him on to stage, I just want to acknowledge Dr. Michael Leon for helping put this together as part of the Illuminations series that the university is coordinating. So without any further ado, let's welcome Dr. Ian Parker.
I think I need that. Okay, can everyone hear me okay there? Turn it on. That would help. Where's the... Okay, is that good now? Yeah. Okay, well, thank you for that introduction, Frank. It's a hard act to follow. So, what I'm going to be talking about, this is part of the Dean's Distinguished Research Series talks. So it's going to be about my research, looking down microscopes, looking at the inner workings of cells. But also this is being held in conjunction with the Illuminations exhibit, showing some of my photography. And what I wanted to begin with is to draw some parallels between those two aspects of my life. To show how my photography can illuminate some of the science that I do. Okay, so let me begin with my title. You can see a lot just by looking. You might recognize this as a saying of the famous Yankees catcher and sage Yogi Berra. And to a large extent that it underlies my philosophy both in doing photography and in research. There are wonderful things out in the world. There are wonderful things to see down a microscope. And a lot of what I do is driven by curiosity, to see things, look at them, try and figure out what's going on. And beginning at a large scale, photography, that can encompass anything from galaxies to strange terrestrial formations, to wildlife. There's lots of beauty to be found out in the world leaving the lab. And in the lab, I don't need to travel down the microscopes. We can see beautiful things. What's shown there are patterns of calcium waves inside an immature frog egg, something I'll be talking about later. So you can see a lot just by looking. Let me draw a parallel between ways of doing science and approaches to photography. Science, the classical way of doing science, is called hypothesis-driven. You have some hypothesis, an idea that you have, something that explains how things work. And in the science, you then go do experiments and test, is that hypothesis right? Strictly speaking, what you should be trying to do is disprove the hypothesis. But usually we have a hypothesis, we love it. When we do the experiments, we are really hoping they'll come out the way that our hypothesis predicts. An analogy with photography, something Ansel Adams, famous landscape photographer, coined pre-visualization. That like a preformed hypothesis in your mind, you have a preformed image of what it is that you want to create. And then you go out into the world with your camera and you try and capture that image. Another way of doing science, observational science. That's where you don't start off with any preconceived notion. You just look and see what's happening. And in the past, observational science wasn't regarded so well. It wasn't regarded as real science. But now there's a growing appreciation that it's a very valuable adjunct to ways of doing science. And it's an approach that I frequently take in my own research. Again, an analogy with photography, saying by another famous photographer, F8 and be there. The F8 there just means that's the setting you should set your lens to. You'll get a good photo. The important part is the be there. You just go somewhere. You don't have preconceived notions what you're going to find. You just go there with an open mind and you look. You see what is happening. So again, let me just illustrate a few photographs to make that point. This is a pre-visualized photograph. These are rock formations in Monument Valley. A couple of days in September every year, the angle of the setting sun casts a shadow of one formation on the other. That's entirely predictable. You can go to Google Earth, see exactly when it's going to happen. Google Earth, you can even work out this is exactly where I need to put my tripod. All I have to do is go there, wait and open the shutter, and I will get the photograph that I have pre-visualized. But an example of just being there, same place, this is Monument Valley, a place that's been photographed so many times it becomes a cliche. But on that time there was this wonderful cloud formation. Not predicted, of course, I just saw it and thought, okay, that's it. That will make a great photograph. So we might come across chance things out in the world, looking down our microscope. We might come across interesting behaviors. Here's a penguin taking a shower bath into a little waterfall. This is just a local shot from Bolsa Chica. Some courting terns, interesting behaviors. And down the microscope, we can also see lots of interesting behaviors. Here we have two amorous terns. Here looking into a lymph node, we have T cells. They're looking a little bit amorous to one another. Interesting behaviors. So we can see things at many different scales. And just going back to this contrast between hypothesis and driven and observational scientist, just emphasize that sentence in the paragraph I showed you. Descriptive observations play a vital role in scientific progress, particularly during the initial explorations made possible by technological breakthroughs. And that applies very much to optical microscopy, light microscopy, that in recent years, the last 20 years or so, there's been a revolution in optical microscopy. For a long time, something like 100 years, microscopes pretty much just looked like that. The early German microscope makers at Zeiss, they figured out how to optimize all the lenses, and for a very long time it's been thought that there were limits to how good a microscope can be. They're physical limits set by the wave nature of light, something fundamental that we could never overcome. But more recently, different types of microscope have been invented. We can't break the laws of physics, but there are ingenious ways to sidle around those laws, to do things that weren't previously thought possible with light microscopes. And if we have a new way of looking, a microscope that lets us see things that we couldn't see before, gives us better resolution, if we have a new way of looking, then very probably we stand a good chance of seeing new things, visualizing things that we couldn't look at, couldn't study before. So for a very long time, I have been building microscopes. It's about 40 years now. These are 3 of the ones currently in use in the lab, 3 that I'll be talking about. I've listed there some advantages of building your own microscope. I won't belabor these are fairly obvious. Let me just say a little bit more about that one. It's fun. When I was a kid, I built robots and rockets and that sort of stuff. So in my scientific career, I've been able to continue doing that. Build ever better toys, and the nice thing is the National Institutes of Health will give me grants funding to do this, to build them. Sometimes, to be honest, there's a new idea for a new sort of microscope, a new imaging technique, and I think that's a neat idea, I'll build one and see what I can do with it. Other times I have a purpose in mind. Okay, so what do these new sorts of microscopes do? The 3 types I'll be showing you, they work in different ways, they have different advantages, they're good for some things, not so good for others. But there's a common theme runs through them, common problem that they're trying to overcome. And again, I'll try and illustrate that by an analogy with photography. And that issue is subject isolation. The world is a complicated place. When you look through the viewfinder of a camera, look down a microscope, there's a lot of stuff there. Stuff that can confuse, get in the way. And for both taking photographs, for microscopy, we want to get rid of all the clutter, to concentrate on just what we want to see. The painter starts with a blank canvas and can put only what she wants on that canvas. But through the viewfinder, through the microscope, there's a lot of clutter there. We want to get rid of it. How can we do that? In photography, one way is to use a camera, a lens that has a wide aperture. It has a big glass at the big front of it. And that sort of lens gives a narrow depth of field. You can focus on a narrow distance away, put everything behind as a blur. So that's illustrated here. We're focusing on that goose. There's hundreds of geese back there. I blurred them out so they don't distract. Microscopes do the same thing. A microscope objective lens inherently has a high aperture. You can see things in sharp focus, where you're focused, but if you have a thick specimen, things above and below will be blurred out. In photography that works. This is not very distracting. Where am I? The blurred background is not very distracting, but down a microscope the specimens we're looking at are rather transparent. You can't see through that goose, but down a microscope your specimen, you'd see the blurry background coming through. What more could we do to get rid of the background? We can try and take it away. This photo, I got rid of the background effectively by photographing these puffins against a black sand beach in Iceland. Black, so you don't see any background. An analogy, if we're looking down a microscope, have a microscope specimen, we could slice it very thin, like a slice of pastrami. We can have a very thin slice, so we're only focused on that slice. We don't have any background getting in the way. That, in fact, is a very good way of looking. If you're looking at a dead, fixed specimen, you can slice it. But what we want to be doing is looking at live specimens. You can't slice them up and expect them to stay alive. Another approach, an analogy for what we actually do in a microscope, is just to selectively illuminate the subject. This is a photograph of a desert lily I took just 2 weeks ago in Anza Borrego during the bloom. Beautiful flower, but it was growing on a patch of very scrubby, dry land that made a terrible background to the photograph. So what I did was wait until night time. Then I asked my wife Annie to hold a little flashlight behind the flower, back illuminate it so I could take a photograph. The only thing illuminated is the flower. The background's gone away, it's dark. And we can do the same thing with a microscope. We can illuminate just where we want to be looking. And the reason we can do that is all the microscopes I'm going to be talking about are fluorescence microscopes. We're not shining light through a specimen and looking at it. We're exciting fluorescence in that specimen. There are many organic dyes, proteins which will fluoresce. You shine something like ultraviolet light or blue light on them, they glow. It's illustrated here. We shine a short wavelength light like UV. We have a specimen which glows. We can put a filter to cut out the excitation light here. We only see the fluorescent glow. So we can introduce into cells different probes, different dyes that fluoresce with different colors. We can identify different components of a cell. But still, we have this issue of sectioning, getting rid of out-of-focus fluorescence. This is a very thin section, so it looks good. But if we're dealing with something thicker that has depth to it, a larger cell or a tissue, then we are focused here. In our microscope, we're introducing the excitation light, a regular fluorescence microscope. That light is illuminating the entirety of the specimen, all the depth. We are focused at this plane. We have our objective lens. We project that focal plane onto a camera. But what we see is a very fuzzy image. We're sharply focused here, but then there's all this blurry out-of-focus background. And I'm illustrating that here with a test specimen. This is a picture of fluorescent pollen grain with a regular fluorescence microscope. I don't study pollen grains. This is just a nice test specimen. It's a pollen grain that looks like a medieval weapon, a flail. A steel ball with spikes on it that knights of yore used to bash each other over the head with. It's a nice specimen for testing a microscope. How well can you see the spikes against the glow of the big ball inside? So this is with a regular fluorescence microscope. We get a terrible looking image. But we can do much better if we selectively illuminate the excitation light that's generating that fluorescence, if we can focus it just where we want. So here's a very simple illustration, a dish with a fluorescent solution in it. We're shining a laser beam. We're only exciting fluorescence where the laser beam is. And by means I'll be showing you, we can do that with a microscope. Here's such a microscope taking optical sections. This is a picture of that same pollen grain I showed you, but it's an optical section, much better. It looks completely black. Why is it black? It's because initially the microscope is focused above the pollen grain. There's nothing to see in that section. But if I play this movie, now we're taking a whole series of sections through the pollen grain. And if I can manage to stop this somewhere, okay, now we're getting a section of that pollen grain. We get a very nice crisp image. A bit like if you go get an MRI or a CT scan of your brain, you can get sections of it. We're not damaging the specimen. All this is being done by light. Okay, so we can take a whole series of sections like that. We can feed them into a computer we can then get the computer to reconstruct a 3D representation. We can look at this thing from any angle that we want. Okay, so what I'm going to be telling you about are three different microscopes. Three microscopes working on different principles. Microscopes I built. Total internal reflection fluorescence microscope. That's a bit of a mouthful, so we just call it a TIRF microscope. A way of just looking at a very thin section at the bottom of a cell. And I'll illustrate that with experiments we've done to look at calcium signaling in cells and how calcium signaling may be disrupted in Alzheimer's. Then a two-photon microscope, a way of looking deeply into intact organs. And with that we've been visualizing the movement and interactions of immune system cells. That's a long-standing collaboration I have with Mike Cahalan over at the medical school. And last, a very new sort of microscope, lattice light sheet. This was an invention that was published only 2 years ago, which I've adopted. And we're using it to look at quite a new biological topic, things with the intriguing name of tunneling membrane nanotubes, thin cytoplasmic tubes that connect cells that are some distance apart. So first, this TIRF microscope. This is the picture of the one I built. It looks pretty complicated, but the actual principle by which it works is quite simple. And let me try to explain it to you in this slide. Here is the cell that we're trying to look at. In that cell we've induced some sort of a fluorescent probe, something that marks the membrane, the cytoplasm, maybe even a fluorescent probe that responds to calcium, so we can monitor calcium signals in the cell. We have that cell on a cover glass. We're looking at it with an inverted microscope, so the objective lens of the microscope is underneath, looking up through the cover glass. To excite the fluorescence, we bring in a laser beam, and we bring the laser beam in at the side of this objective lens, so it goes at a shallow angle through the cover glass, and when it hits the boundary, the edge of the cover glass, between the glass and the aqueous medium inside the cell, the fluid, the medium it's bathed in, there's a refractive index difference. This laser beam undergoes total internal reflection. You may remember that from high school, and if that's all that was happening, it wouldn't be very interesting. This light has just bounced off the cover glass. But what happens is the light penetrates a tiny distance beyond the cover glass. Only about 100 nanometers. To put that in perspective, it's about 1,000th the thickness of a hair on your head. So we can excite fluorescence in a very thin section right at the bottom of a cell, next to the membrane of the cell where it's contacting the cover glass, and just inside the cell. So it's giving us an optical section. Anything that's glowing deep in the cell we don't see. We're only looking at what's the bottom. This is illustrated here. Cell expressing a green fluorescent protein. By conventional fluorescence microscopy, you get a blurry image. You're seeing the fluorescent protein everywhere through the depth. Here's the same cell, imaged by TIRF. You're just seeing what's down at the membrane at the bottom of that cell. And we've been using this technique for many years to look at calcium signaling in cells. So let me just give you a little bit of background. What is calcium signaling? Calcium is important in our body in two contexts, very different. We're told to drink lots of milk to get lots of calcium. That's for structural elements, the bones and teeth in our body. Inside cells, calcium is used in a completely different way. It's used as a signal, a messenger to send information within a cell. And changes in calcium inside a cell, in the cytosol, they regulate numerous important functions in just about every cell in our body. How does it work? Inside a cell, concentrations of calcium are maintained at very low levels. Outside a cell, there's lots of calcium in the extracellular fluid, and there's lots of calcium in intracellular organelles, particularly the endoplasmic reticulum. Calcium inside the cell, it's about 10,000 times lower than outside or in the ER. So there's enormous gradient. Calcium wants to flow into the cell. The membrane of the cell, the membrane of the ER, normally doesn't let it. But those membranes have channels, proteins that can form a little pore, a little pore that can be open and shut in a controlled manner to allow calcium to enter in a very precisely controlled way. And these elevations of calcium inside the cell, they're a signal. They control vital functions in every cell. Some examples: contraction of heart and skeletal muscle, secretion of saliva, synaptic transmission between the nerve cells in our brain. That's all done by calcium. Calcium does many things. In some cells it can do two things, two different things. A wonderful example is in smooth muscle in the gut. There, if levels of calcium locally go up very close to the membrane, they produce an electrical signal that causes that muscle to relax. But if calcium, the same thing, if it goes up inside the cell, it causes contraction. So calcium, the same messenger, giving two completely different results. How's that possible? The answer is spatiotemporal patterning of calcium signals. Where in a cell is the calcium going up? For how long does it go up? And this is a topic I've been interested in for many years. How are the patterns of calcium signals in a cell generated? How are they generated under normal physiological conditions? How can it go wrong in disease states? Now, let me give you an example here. I'll leave this movie playing. It'll probably distract you while I explain what's going on. Here we're imaging a giant cell, an immature egg of the frog. It's about 1 millimeter across. We're looking at the whole cell. What we're looking at is calcium inside that cell. The cell has been injected with a fluorescent dye, a dye whose fluorescence changes with calcium. It glows more brightly when levels of calcium go up. And here we're color coding that. Higher calcium, more bright fluorescence is a warmer or redder color. What we're looking at is calcium being released from the store, the endoplasmic reticulum. There are channels in the ER that release calcium into the cytosol where we are looking. Those channels, they are controlled by another signaling messenger molecule, inositol trisphosphate, IP3. This is generated by things like hormone, growth factor, and neurotransmitter receptors on the surface of the cell. But here we're just artificially elevating IP3 in the cell, opening channels in the ER membrane, getting calcium released into the cytosol, and as you can see, that's producing beautiful patterns, waves of calcium sweeping across the cell. So this was an early experiment. I've taken this from Jim Lechleiter, who has a better movie of this than I do. But this was just taken with a regular fluorescence microscope. I was interested in looking finer details, what's going on to generate those waves. So then with Angelo De Muro in my lab, we started using a TIRF microscope to look at finer detail at calcium signals. And what I'm showing you here is a movie. We're looking at the same cell, the oocyte, but we're zooming in. We're looking at just a small region, and we're looking with better resolution in space and time. And what we see then is two different types of calcium signal. There are little volcanoes, little local eruptions of calcium going on, things are called calcium puffs. And if the movie hadn't frozen, yeah, then we see a calcium wave. Let me play that through again. Look at the top left-hand corner. You see a puff went off. The next time it goes off, if this computer would stop freezing up, okay, it triggers a wave that sweeps across that region of the cell. So we discovered two types of calcium release. The wave, which can sweep across the cell, a global signal, and little local signals, these calcium puffs. And the question I had was, these little local signals, the calcium puffs, are they released through individual channels in the ER membrane, an individual protein that's opening up, or are they concerted opening of several channels all going off together? So over time we improved the resolution of the TIRF microscope and tried to get it down to the level where we could resolve single channel signals. First method of recording from individual ion channels in the membrane, a technique called the patch clamp, invented by Erwin Nair and Bert Sackmann. They came up with a technique to electrically measure current flowing through individual channels. As the ions went through, they generated electric current. They were able to measure that with a little glass micropipette that they pushed against the membrane of a cell. And what they recorded were little square pulses of current here, corresponding to the opening of individual channels, individual protein molecules opening to allow a flux of ions. This is one of their very first recordings. It was published in 1976. I still remember opening the issue of Nature where they published that paper, reading it. I had two reactions. The first was, wow, this is fantastic. This is the first time we've ever been able to record the activity of a single protein, a single ion channel in a live cell. My second reaction was, damn it, why didn't I think of that? Because like many of the best discoveries, inventions, once it's explained to you, yes, it's fairly simple. But I think that thought stayed with me for a long time. Yes, 30 years later, with Angelo, we were recording from exactly the same type of channel here. In our case, we'd expressed it in the membrane of the Xenopus oocyte, but we're recording from channels not with an electrode, we're not recording currents, we are recording optically with our microscope. We are recording tiny amounts of calcium coming through individual channels. This is one of our tracers. I actually think it looks better than Nair and Sackmann's first trace. We get a better resolution. Of course, the patch clamp, it's got better in the intervening years. So two advantages of doing this optically with a microscope. One, we can look at a patch of membrane. We can see a little fireworks display going off. All of these little dots, these flashes, they are individual channels opening. For each of them, we can measure what is the fluorescence as a measure of the calcium coming through. We can get traces like that. We can record the activity of many, many channels simultaneously. And because we're doing it optically, we can look at channels inside a cell. We can look at channels in the ER if they're close to the cell membrane. With the patch clamp, you have to press an electrode against the outside of a cell. You can't easily look at what's going on in the internal membranes. So we use this technique. These are data from Ian Smith in the lab. Now we're looking at the calcium puffs. We're not looking at single channels in the membrane, we're looking at release from the endoplasmic reticulum. And if this little movie will play for me, it'll look very similar to what you had before, little flashes, but now these are the calcium puffs, release of calcium from the ER. Again, we can get traces from each of these sites. I've just lined up several of them here. And you can see we're now getting events, calcium signals of differing sizes, but some of them look just like single channels, little square openings. So we think what we're seeing here is these signals are individual channels opening. These larger events, the calcium puffs, they have steps on them, and we think this reflects the openings and closings of individual channels. So these calcium puffs that I showed you, they're the concerted opening of several channels going off together. So these observations let us produce a hypothesis, a model for what we think is going on in cells. A model that we originally derived looking at the oocyte as a model cell system, but we now know it applies to many, many different types of mammalian cell. We can have signals where just one channel opens, a little square pulse. I don't think I was feeling very imaginative when we discovered these. I call them calcium blips. The calcium puffs, we think one channel opens and then it sets off several channels around it in a cluster. We think these channels, these IP3 receptors, the channels that are opened by IP3, they're clustered in the membrane of the ER. And a property of these channels, they require IP3 to bind, but they're also activated by calcium itself. When one channel releases calcium, that can feed back, it can promote the opening of adjacent channels. So we think here one channel opens, that calcium diffuses a little in the cytosol, opens adjacent channels, a handful of them go off, we get a puff. If we stimulate more, give more IP3, sensitize the system, we have a puff. Calcium from it can diffuse to the next little cluster of channels, set them off, they generate a puff. That calcium diffuses, sets off the next cluster, and so on. We have a wave of calcium jumping from cluster to cluster. So this hierarchy is now generally accepted as physiological phenomenon in many different cell types, and refer to these different signals as they're like the phonemes of the language of calcium signaling. So that's the normal functioning, but it has to be very tightly regulated. What can happen when it goes wrong? My analogy, the store of calcium outside the cell is like the water in a reservoir, the membrane is like the dam, the channels are like a spillway which is releasing that calcium into the cell in a very carefully controlled manner. But what happens when the spillway breaks? What happens when the integrity of the membrane breaks down so we can have an uncontrolled influx of calcium into a cell? And one aspect we've been looking at in that context is Alzheimer's disease. Coined this term a calciumopathy, a disease where there's a disruption of calcium signaling. Just a quick primer on Alzheimer's disease. Characteristic feature is the formation of plaques in the brain, amyloid plaques. You can see them in post-mortem sections of brains from Alzheimer's patients. These plaques are being formed by amyloid proteins, small protein, 42 amino acids wrong, which is being improperly cleaved from a precursor protein. These beta amyloids, the monomers, they're sticky. They clump together. Eventually they clump together to form the plaque, but intermediate stages, you can get oligomers, just a handful of these beta amyloids stuck together, then they stick together to form fibrils, eventually form the plaque. The plaques at one time were thought to be perhaps the major source of damage during Alzheimer's, but now there's a growing view that it's these intermediate stages that are important. And we see a role for them in disrupted calcium signaling. So just look at this trace. What we're doing is recording calcium. We've taken these cells. These are a neuroblastoma cell, a neuronally derived cell. We put an indicator inside a fluorescent dye which changes its fluorescence in response to calcium. And we're recording. I can play the movie, I hope. We apply monomers, nothing much happens. We apply fibrils, maybe just a little signal, but then we apply the oligomers. Wow, massive calcium influx. And that's just plotted on the graph. The monomers, the fibrils don't do much. The oligomers saturated. Enough calcium came in that it very rapidly killed these cells. What's going on there? What might be the mechanism? One mechanism that had been proposed was these oligomers act on ion channels that are normally present in neurons, the channels that would respond to neurotransmitters, that the oligomers might activate those improperly. To test that idea, we went back to the xenopusoocyte. It's an egg cell. It doesn't have the channels that a neuron has. So we were putting the A-beta oligomers on xenopusoocytes with a TIRF microscope looking. And what we saw, let me play this movie, we saw lots of little flashes. Calcium coming through the membrane, through distinct little pores. We don't think the oocyte has these channels there to begin with. What we think is the oligomers are aggregating, they're inserting into the membrane of the cell. They themselves are forming little calcium permeable pores. It's almost as if they're punching holes through the cell membrane, letting calcium come in. And this is just a map of all these little sites in the membrane, massive number of them. If we look at the fluorescence from individual sites and measure the calcium coming in there, we see records that look very much like our channel record. So we think these oligomers are forming little pores, channel-like pores, but allowing an uncontrolled entry of calcium. And that may be at least a contributory factor in Alzheimer's disease. A small amount of calcium coming into neurons is probably going to subtly disrupt synaptic signaling, but if too much calcium comes in, ultimately it will directly lead to the death of those cells. Okay, that was one theme, one type of microscope. TIRF microscope, wonderful for looking right at the bottom of a cell, but you can't look into a cell, you can't look into tissue with it. Different sort of microscope I've been using is a two-photon microscope, something that allows us to see deep into intact tissues. How does it work? Let me just go back to the basics of fluorescence. Regular fluorescence, we have some molecule which we can make to fluoresce. We excite it by shining a short wavelength light on it, blue light say. The energy of that blue photon kicks this molecule into an excited state, and very soon after it decays back to the ground state. There's a little loss of energy in this process. The photon that gets emitted is a lower energy, shifted more towards the red end of the spectrum. So we excite with blue, we get a green fluorescence. Two-photon excitation, the difference is that now we excite the fluorescence not with a high energy blue photon, but with two infrared photons. Deep in the red, these photons have a half the energy of this blue photon. One of these alone couldn't excite fluorescence, but if we can hit this molecule at almost exactly the same time with two photons, we can excite it again, and it will drop back to the ground state. It will fluoresce just the same as with one photon excitation. Okay, how can we do this? Why is it useful? Okay, the doing of it takes some trick. This is a process that never happens in normal life. You need an incredibly high density of photons, incredibly intense light for this to occur. These two photons have to hit at almost exactly the same time, and in this context, almost exactly the same time means about 10 to the minus 19 of a second. So how can we make it happen? We can make it happen using a microscope. Here is the objective lens of a microscope. We're focusing light down. Here's the focal point. All the photons coming in, they effectively get compressed at the focal spot. So at this spot, we have a much higher density of photons. Maybe we could make two-photon excitation occur here, but it wouldn't occur above and below. We can get our selective excitation of fluorescence. That's illustrated here. This is one photon regular fluorescence excitation, the light coming from the objective. It's exciting fluorescence all the way through this hourglass shape. Two-photon excitation here. We're only getting fluorescence, that tiny little green glowing dot. It's only at the focal plane. There's nothing above and below. We got rid of out-of-focus fluorescence. That's great, but there's one more trick. If we were to do this with a continuous light, the intensity we would need would be such that at this focal spot, our specimen would instantly vaporize. The light would be incredibly strong, and the trick to making this work is using a special laser, a laser that generates incredibly brief but incredibly intense pulses of light. During each pulse, the intensity is enough to give this two-photon excitation, but the pulses are spread out, so averaged over time. The laser exposure of our specimen is quite modest. We don't do it any harm. So we have a little spot. We can make an image by scanning this spot around our specimen, build up an image point by point, pixel by pixel. And there's one more advantage of this technique. We're getting a section, in effect. We're using infrared light to do it. Infrared light isn't very much scattered by biological tissue. It'll pass through biological tissue. Blue light gets scattered. So with infrared light, we can see deeply into our specimen. Okay, so what can we use a two-photon microscope for? I built one. I intended it to use it for imaging calcium signals in the brain. But one day, Mike Cahalan, a colleague, knocked on my door and said, "Hey Ian, I hear you built a two-photon microscope. Could we look at a lymph node with it?" The story I have at the time is that I'm in a neurobiology department. I have colleagues who work on the brain. If you'd asked me what the most boring organ in the body was, probably I wouldn't have even thought of a lymph node. But it turned out to be fascinating. We put fluorescent T cells into a lymph node, looked under the microscope. Lo and behold, we can see T cells inside the lymph node. We warm the lymph node, give it oxygen, the T cells start swimming around. One of the topics we were looking at was the immune system. How are the T cells activated in response to an infection? Let me just quickly talk you through this. You get a wound in your skin, bacteria come in, there are cells out in the skin patrolling, cells called dendritic cells, they're the green guys here. They pick up proteins from the bacteria, they chop up those proteins, form little antigens, and the dendritic cells go back to your lymph node. And there they have the job of presenting this antigen to T cells, which if they recognize that antigen, the T cells will divide, they'll go off to fight the infection. But there's a problem there, that we have over 1 million different sorts of T cells, each is specific to a different antigen. And there's a needle in a haystack problem. How does any given T cell specific for a particular antigen, how does it find the dendritic cell presenting that antigen? What we're looking at, it's a process called repertoire scanning. How do the T cells scan the dendritic cells? So with our imaging, we're able to look inside the lymph node and see what's going on. Here, we've put a fluorescent dye, a green fluorescent dye to label the dendritic cells. The T cells are red. And if this movie will play well, okay, things are remarkably dynamic. There's lots going on. And you can see immediately, just looking, two processes that are going to enhance the scanning. First, the dendritic cells, the DC, they have the processes, dendrites, they are madly gesticulating. They're waving around, they're saying, here's some antigen, come find me. The T cells, they're all buzzing around. If we actually track what they're doing, we can plot their tracks. They're doing a random walk. They don't know where they're going, they're just randomly walking. But computer simulations show this is actually quite an efficient way of scanning, getting T cells to encounter the dendritic cells. So in this movie, we haven't had any antigen present. This is just the default scanning mode. This is going on right now in your lymph nodes. All the T cells are buzzing around, the dendritic cells are waving around. What happens if we actually give an antigen? Here, we've created an artificial situation. We've induced in the lymph node many T cells, red labeled, but all of those T cells are specific for the same antigen. And we've presented that antigen, the dendritic cells are presenting it to the T cells. And if I play this movie, you can now see a different sort of behavior. Now the T cells are swarming. They're swarming around the dendritic cells. They're recognizing that cognate antigen. They're swapping chemical signals with one another. These T cells become activated. They go off, they divide. If you look on the right, look at this cell, this rather enlarged one. It is about to divide. I play the movie. You can see the cell division, the two daughter cells. They look as if they're interacting for a bit, then they go off. This division process will continue. There'll be a big multiplication of the T cells. They can go off and help fight the infection. And one thing T cells do, or a particular class of T cells, when they're activated, they can give help to B cells. B cells are the lymphocytes that are going to generate antibodies. And there's a problem there because the B cells in a lymph node, they're in a different neighborhood to the T cells. This is a section of a lymph node. Here we've got red fluorescent B cells, green fluorescent T cells. They're in different places, but they do meet up at the edge of this follicle. And when we have a cognate antigen, activated T cells and B cells, the B cells actually move towards the edge of this follicle and they link up with the T cells. Let me play this movie. At the edge of the follicle, they pair up. The B cells pair up with T cells and they do a waltz. They're waltzing around, the B cells are in charge, they're leading, the T cell is following behind. Mike has a nice video of this. He set it to music, Chopin. That's appropriate for the B-cell, T-cell waltz. Okay, last thing I'd like to describe to you, again, a different sort of microscope, a different sort of biological topic that we've been studying with it. This is called a lattice light sheet microscope. This is an idea that was invented by Eric Betzig. Betzig won the Nobel Prize it was 2 years ago now for a form of super resolution microscopy, seeing individual molecules. But about 2 weeks after his Nobel Prize was announced, he had a paper published describing this very different sort of microscope, lattice light sheet microscope. And he said, this lattice light sheet microscope, it's better than the one I got the Nobel Prize for. So I thought, hey, I better try that. Let's see if I can build one. Okay, it's quite complicated. I won't try and explain how it works very much in detail. It took me months to get the damn thing working. But last year I got a working lattice light sheet microscope. A little while after that, the first commercial version became available. You can now buy one like this. It doesn't look terribly different from mine. One difference is it'll cost you $800,000 to buy one of these. Mine cost about $80,000 in component costs, plus bits I had lying around the lab anyway. Okay, so in simple terms, what is a lattice light sheet microscope? How does it work? Again, it's a way of getting an optical section, getting a thin slice through a live cell, imaging it by fluorescence. The way it works, instead of just one microscope lens objective, there are two. Here is our cell. One objective lens is projecting light through the cell. It's projecting that light as a very thin sheet of light, very thin this way. Second objective lens, we're looking at the fluorescence which is excited in this sheet of light. So again, we've confined the fluorescence to just where we're focused, just on this sheet. There's no fluorescence being emitted here or here. This basic idea, light sheet microscopy, it's been around for quite a while. But the trouble has been creating a very thin sheet which extends over some distance. That was Betzig's innovation, a way of generating a very thin sheet of light which extends right through the thickness of the cell. And the essence of it is the optics in the microscope create a lattice, an array of very thin rods of light. These things are called Bessel beams, very thin rods of light that go through the cell. And let me play this movie. That rod of light, it's dithered. It's quickly moved back and forth to even it out. So what you end up with is a thin sheet of light going through your cell. At any given time, you're only looking at one sheet, but you can move the cell back and forth through that sheet. Again, you can capture a series of sections. You can get the computer to regenerate a three-dimensional image for you. My original intent in building this was to look at calcium signaling again. But we've got very much involved. Ian Smith in the lab is looking at these things called tunneling membrane nanotubes, TNTs. We have two cells, they're well separated from one another, but joining them, there's a thin membrane tube filled with cytoplasm. And this is both a physical bridge and it's a way of communication between the two cells. We can get signals passing from one to another. In a minute I'll show you calcium signals passing from one to another. These TNTs, they're becoming identified in different disease states, maybe a way that diseased cancer cells can communicate with one another. It's even been proposed that organelles, mitochondria, can move from one cell to another. Though we haven't seen it, I have a little bit of doubt as to whether that's actually a real phenomenon. But whatever, a novel way of communication between different cells. Studying these things has been difficult. These tubes are very, very thin, they're long, they're very delicate. The light that you use on your microscope to image them, it can destroy them. But it turned out the light sheet microscope is a very good way to image these tunneling nanotubes. So Ian and I have been using this to look at tunneling nanotubes in a cancer cell model. Breast cancer cells that have a propensity to go to the brain, to metastasize, to form brain tumors. So looking in culture, growing these cells in a culture dish. Here are some images. Here are some cancer cells. This is reconstruction, a 3D reconstruction from a series of light sheet images. You can see these nanotubes. Some of them seem to lose their way. They just stick on the cover glass. But some here, they are contacting between this cell and those cells. This is a side view of the same thing. These tunneling nanotubes, they're suspended up in the bathing medium. They're not going along the cover glass. I liken them to the strings of a harp. They're stretched tight between these two cells. And I can give you a better illustration here, this movie, 3D reconstruction. Same cells here, but now, if this video would keep on going, we can spin these cells around, look at them from different angles, and at the end, this is now a time lapse. When the spinning stops, we do a time lapse over about 40 minutes. You can see there are things going along the nanotubes. We've labeled the membrane with a fluorescent protein. You can see membrane components moving along the nanotubes. And these nanotubes, as I said, they can communicate signals. Here are two cells. Here we're looking at a fluorescent protein in the cell membrane, so we can see the TNTs connecting the two cells. On the right, we've loaded these two cells with a fluorescent indicator dye, something we can use to look at calcium levels. These are the same two cells. Let me play this movie. We stimulate the top cell to generate a calcium signal with a little spot of laser light. We get a calcium signal. Several seconds later, this cell responds. We've got a message, information passed from there to there. We think it is this messenger IP3 going along the nanotubes. And here again, transport of membrane constituents. If I play this one, you can see things in the nanotube moving. But if this video would just keep going long enough, the really interesting thing, there's a vesicle floating around. It comes in, sticks to the nanotube, and then it looks as if it's hitchhiking. It's a sort of FedEx for delivery of extracellular vesicles, a new mechanism of communication. Okay, so this is all very new. It's still at the very beginning. What I've shown you up to now is in cell culture, which is where most people have studied these tunneling nanotubes. They're much easier to see there. But because they've been studied primarily in cell culture, there's always been skepticism. Are these things actually present in tissue? Are they doing something? So we've recently been looking in brain tissue. We put human breast cancer cells into a mouse, an immunocompromised mouse. Then some weeks later, euthanize the mouse, take sections of the brain, look at them with our microscope, our lattice light sheet microscope. And most clearly here is a series of sections, cancer cells, fluorescently labeled in brain tissue. And you can clearly see tunneling membrane nanotubes. They are within a brain tumor. So at the moment, this is very early days. We don't know what they're doing, what their role might be. We're at the looking stage. We're seeing interesting things. From then, we learn more. Ultimately, we go on, make hypotheses, try to figure out what the mechanisms are, how this may be involved in the progression of tumors. Okay, so to finish, just to acknowledge the people. These are the folks in my lab. Angelo De Muro there was involved in the oocyte work, developing this optical patch clamp technique for imaging individual cells. Ian Smith, he was involved in a lot of the PUF imaging work, the TIRF, and now most recently in the tunneling nanotubes. Kyle, a great graduate student in the lab, he does the software for us. He's written the software for controlling our lattice light sheet microscope, for analyzing our images, for analyzing our calcium signals. Mike Cahalan, of course, we've been collaborating for, I think, about 16 or 17 years now, looking at the immune system. Started just from curiosity, but we began a whole new field of immunoimaging. Many immunology labs are now using two-photon microscopy to look at the immune system. And Mark Miller, who then was a postdoc in Mike's lab, spent many days in the dark in the two-photon looking at T cells crawl across the computer screen. And last, just to acknowledge my wife, my companion, on all our travels around the world, photographing things. She refers to herself as the photographic elf. She drives me in the cold, dark morning to wait for sunrises. She brings me the tripod and just generally looks after me. So thank you very much, Annie.
So we have time for a few questions for Ian.
Larry? Ian, I'm struck by what seems like a parallel between your puffs and the famous end plate noise, otherwise known as MEPS, the kind of stochastic background, the quantal nature. Is that more than a coincidence?
I think it's a coincidence, but it's a very nice coincidence. I'll give you a little bit of history on that. When I was back in London, I was in the biophysics department, which was chaired by Sir Bernard Katz, the person who discovered things he called MEPS, little spontaneous blips, events, electrical signals in a muscle cell. And I felt when we discovered the puffs, hey, we're sort of recreating history. It's a very different mechanism, what's going on, but again, little quantal unitary events. And I was very happy one time. Bernard Katz died several years ago, but I went back to University College London. I met with him. I showed him our paper on the calcium puffs, and I said, you know, I feel like I'm walking in your footsteps. We're recreating history in a very different sense, but little quantal events.
But you're sure there's no parallel underlying mechanisms there? It's more than just a coincidence?
I think the underlying mechanisms are very different. The MEPS, the electrical signals he recorded at the muscle, they are due to the release of vesicles of neurotransmitter, about 1,000 molecules of neurotransmitter packaged in a vesicle which is released. What we're seeing is calcium. It's calcium coming through channels inside a cell. The mechanism is really quite different, but still there are parallels, that in both cases the signaling is in a sense quantized. It's coming in little packets, not as a continuously graded fashion. Yeah, you have a question? Or maybe you were taking a photo with an iPhone. Oh.
The
go ahead, Larry.
nanotubes are fascinating. Are they purely pathological? Do they only exist for pathological purposes?
We don't know. We see them between these cancer cells. We see them between cell lines in culture, but then of course we are talking about immortalized cell lines, which are cancerous. As far as I know, they have not been seen between what you could say is normal cells, but maybe we haven't looked enough.
I'm intrigued by those nanotubes, and what came to my mind is it possible as a hypothesis that they can be examined as a communication cutoff to growth of tumor cells? I mean, if they're communicating with one another, hey, let's get bigger and cause damage, what if we figure out a way of really examining those tubes to see if they can be, the communication to be cut off for the growth not to happen?
Yes, there are certainly possibilities of the nanotubes being a therapeutic target. There is one paper that's been published knocking down the formation of these nanotubes in tumours and showing that that makes the tumours less resistant to chemotherapy. So again, this is very early days. We've only started working on this problem just a few months ago. It's still a new field in the biology, but I think it has great promise as a potential therapeutic target.
My question is more general. So of all the amazing discoveries that you've been a part of, what is the coolest thing for you that you have seen both through the camera lens and then down a microscope?
Okay, that is a difficult one. Okay, down the microscope, I think the biggest discovery I had was these calcium puffs. There was something, I thought there might be something like that, but to actually see them, down the microscope on the computer screen, that was a revelation. It's one of those times. Science usually is a very slow process. Most of the time things go slowly, most of the time things don't work. But very occasionally you have this day when you see something and wow, you dash out of the lab, you go down the hall, try and find people to pull them back into the lab and say, hey, look at this is wonderful. So I think the discovery of the calcium puffs, well, that's probably the highlight so far of my career. And photographing side, I'm not sure I can say one photograph stands out. Let me tell you the favorite place on Earth for photography, South Georgia Island, a little island in the Southern Ocean just north of Antarctica. We go there, you land on a beach, and literally there are about 300,000 penguins on that beach. You just sit there, they will come right up to you, you're just immersed in them. So I think as the most outstanding experience in my photography, that counts.
Okay, Peter, you have a question?
Yeah, that was a lovely talk. How long did it take you to build the light sheet microscope? If you had to build it again, how long would it take, and what would you charge for it?
Okay, so the first one, I don't know, it was over a year to put together. That's not a year full time, of course. This is just going in, tinkering with it. It was a slow process. It was a learning experience. It is a difficult thing to build. A two-photon microscope is trivial in comparison. One night I was having trouble falling asleep. To get me to sleep, I thought through how many individual adjustment knobs are on that microscope. There are 52. All of them have to be exactly right for it to work. So first time it was a learning experience, it took a long time. I think now, if I were to build one, I could probably put one together in a few weeks. I know what I'm doing. If I'd ordered in the components, everything was ready, I think I'd have it going in a few weeks. Okay, how much would it cost? Okay, that's the art of the deal. We need to get together and talk about that.
So why don't we thank Ian for a wonderful talk. Ian, thank you for this.